So, is there a laser pointer here? Sorry, perhaps here. Yeah. Okay. So today, what I want to tell you about is really what I a corollary of my work as a biologist, as a neuroscientist. What I want to talk today about is, our proposal to perform neural morphometry directly from bitmap images. And why is this important? Um, so as you all may know, neural functions is intimately related to neuronal shape. So in order to understand how neurons work, one needs to be able to quantify their anatomy. So anatomy of neurons is critical for classification of neurons, to understand their physiology, their connectivity, and neurodevelopment, which is mainly my area of expertise, and in order to be able also for physiologists to establish computational models of neuronal function. So the problem of all these is that in order to obtain relevant quantitative information from the anatomy of a cell, one needs to reconstruct such anatomies. And this is really a century-old constraint in the field. So, as we were doing, 100 years ago, as it is now, we still need to reconstruct, data from bitmap images. And these, while some years in the beginning we were doing this by hand, we are now doing it semi-automated, but it's still not possible to do it fully automated. And so, just to illustrate the example or the problem per se, I wanted to provide you just some examples of two of the most complex neurons that exist in nature. So here on the left, we have Purkinje neuron from a mouse cerebellum, and this is one of the biggest cells in the mammalian brain. And on the right side, you have a nociceptive sensory neuron from that exists in embryonic, in the body wall of embryonic fruit fly. And these neurons, just to give you an idea, if you need to reconstruct these cells, it would take you about, for an expert, anatomist, something between 5 to 10 hours per cell, or even more. And so, so the problem, since I work with these cells and been thinking really about it, is about how to solve these. And so, what we thought is that at the time where fully automated systems for reconstruction remain unavailable, perhaps other approaches could be implemented in the interim. So what can we do in the interim? Are there other techniques that allow us to extract, anatomical data from images directly that bypass reconstruction? And so, one technique that existed already and that I've been using it for a while is called show analysis. And so, just to give you an overview, what is show analysis is actually the first successful attempt to quantify neuron arbors, and it was established by anatomists at the UCL in 1953, more than 50 years ago. And I won't tell you the rationale behind the technique. I'll just, but although it's a fascinating story, and if you have time, perhaps you can ask me about it. But I'll tell you a bit of how it works. So basically, what Donald Scholl did at the time, he basically drew several concentric shells around focal points in the arbor, and by doing so, he counted intersections between that neural processes were establishing with those shells. And what he found is that by doing that, he could establish relationships between the number of intersections at in function of a distance from those focal points, and he was able to characterize. So, for instance, in the example from his original publication, he was able to characterize the morphology of the basal arbor of a neuron and distinguish from its apical. And while in the beginning this was just an attempt to quantify neuronal shape, we now know much more about why the technique works. And just to give you an overview, a summary, we know that it's actually what it's doing is actually an optimized surrogate for sampling dendritic mass. And so what the field was not, what is not really implemented in by neuroanatomists is that neuroanatomists remain unaware that the technique could actually become fully quantitative. And for a while now, people have been using it in a semi-qualitative manner. And so what so what we have done, and us and others, we were able to make the technique much more robust numerically. So I'll give you an example. So this is the class 4 neuron I showed you before. And so, so this is how a sample data would look like. Again, the number of intersections, the functions from a distance. But if you perform a couple of curve fitting steps and data normalization, you are actually able to establish from samples, you have to modify your sample data in a way that it makes it much more informative. So in this case, we have a heuristic mechanism to establish the best fit polynomial, and we are able to detect, certain features of the profile, such as its maximum, the distance where it occurs, the center of mass. We can also extrapolate the number of primary branches, which is important in anatomy, and establish arborization index. And this is based on the initial sampling distance. And we also can normalize the data to establish linear relationships and extract constants that we call, in this case, the actual decay, which is a measure of how fast the morphology is changing in the cell as it grows. And so I won't go into detail, but we're able actually to establish right now between 20 and 40 metrics that are based only on the show approach itself. So what I did and what I wanted to show you today is the show analysis plugin, which is again based on a technique that I've showed before by Donald Scholl. It does not require previous tracing of the arbor, although it can be used. It can also incorporate data from previous traced cells. And again, it offers you a vast repertoire of metrics for direct statistical comparisons between arbors, which was, I think, missing from the technique. And it's fully integrated in ImageJ, distributed in Fiji. So if you are using Fiji, you already have it in the analysis menu. And while in development has been already cited by several publications, and it's open source. And I wanted to tell you that it's actually, I think, thoroughly documented. So lots of effort was made in the documentation, not only about the function of the plugin, but also about the concepts behind the technique and some of the relevance of these metrics. So just to provide you an overview, so the plugin is extremely simple. So it takes an image, either a 2D or 3D image. The image needs to be segmented, and then the algorithm for sampling data is extremely simple. We are just monitor pixel connectivity at equidistant sites. We do have a sophistication in it that allows to mitigate incomplete fillings, which is the major problem when you're trying to reconstruct really thin neuronal processes that in arbors that span for several distances. And then it has a module that performs all the computations, so curve fitting, data memorization, descriptive statistics. It also allows, again, as I said, to incorporate data from simple net tracers, so the framework in Fiji to reconstruct cells. And then it extracts metrics and masks that then allow you to make statistical comparisons between neurons. So how accurate is it? Does it work? And so to address this, the first experiment that I did was to establish, so I decided to go about this using a very perfect field cell in which an image is characterized by high signal-to-noise ratio. So this is an intracellular field, a pyramidal neuron imaged with a 2-photon microscope. And so, and these neurons are characterized by basal arbor, dendrites here, and the apical arbor here. And the basal compartment is actually has two types of neural processes, as both dendrites and axons, here in magenta, in the reconstructed tracing. And so, as you can see, like in the apical arbor, the apical part here, there is a perfect agreement between the two approaches, while in the basal, of course, the plugin cannot distinguish between axonal processes and dendrites, and it oversamples as it integrates information from both. However, because axons are much thinner than dendrites, it's actually possible in a very straightforward manner to segment the image so that we can discard information from the axons. And if you do so, which is the dark dots here, you can actually bring back the accuracy to the tracing levels. And so we did this across multiple cells. And so here is just a Turing mean difference plot just to allow us to quantify the bias in our approach. And so what we're doing is pairwise comparisons between the two techniques, and these sampled here are at every distance that we've sampled for 3 different cells. We average them, and you can see here that the bias that we have is around 1.5 intersections overshoot over the mean, so roughly 1.5% of the data. So overall, this suggests it is quite accurate. So, but what about images that are not as perfect as that one? What about images that have low signal-to-noise ratio? So the way we decided to do this experiment was to take an image that would be available to everybody so that everybody could replicate this data. And we decided to go about a data set from the diadem challenge that I'll tell you later on a bit more. And the importance here again is that these cells are very well annotated, they are publicly available, and they are associated with the ground truth tracing that we can use for comparisons. So, and then we degrade this image in a controlled matter so that we can make, correlations between the amount of degradation that we've done with the performance of the plugin. And then we segment the image, and then we extract the metrics. And for these here, we have a reference segmentation that I won't have time to tell in detail, but it's described in the manuscript, so you can have a look at it. And so to make to give context to the comparison, what we decided to do also, we said to compare, while we were doing this simultaneously, to use, an approach that tried to reconstruct the neuron automatically. And so we used neural circuit tracer, which is one of the finalist algorithms of the diadem challenge. And so we performed the same analysis. So we performed automated segmentation by neural circuit tracer. It's a great program, by the way. And we obtained the profiles from the tracings, and then we feed them in the plugin. And so, just to give you an idea of the data that we picked, the data set that we picked is an axonal arbor from Drosophila. So in this case, it's labeled by a clonal technique called Markham. And so this is how the original image looked like. And then we perform iterative degradation. And we use the stack standard deviation as a probability mass function. So you can see here in increments of additive noise, okay, to the right, and negative noise to the left, which is perhaps relevant for multiphoton systems associated with loss of signal there. And so just to give you an idea, here we have the correlation of the degraded image with the original one. So the original one would be a perfect match, would be 1. And highlighted here are the two extremes, just for you to have an idea, to grasp how the degradation was performed. And so now I'm going to tell you the result about the experiment. It's going to be a rather crowded slide, but hopefully I'll be able to guide you through it. So here on the x-axis, what I'm showing you is the noise. So again, additive to the right, here to the left, negative. And I'm confronting it with plotting it against the metric that we obtained. And so in the dark dots, we have our results, and then you have the neural circuit tracer, so the automated approach for reconstruction here, and the dashed lines refer to the ground truth. And so, and by the way, I'm forgetting, this is the correlation coefficients that tells the agreement between the two variables, how well they agree between the two. And so we performed this for all the roughly 20 metrics that we are capable of extracting. And again, it's rather crowded, but the slide, but the take-home message here is twofold. So, one is that metrics are actually they are in a good agreement within a certain range of noise. So there's agreement between them, so suggesting that they do indeed resilient to noise. And as you can see, that's highlighted here in the shaded graph, in the shaded area of the graph. And the other one is that the metrics that we obtain from fitted data is actually much more stable than when compared to the ones in the respective metrics obtained from sample data. And perhaps the biggest extreme is the maximum obtained of dendritic intensity, in this case, sorry, axonal intensity. So this is an axon here, and the sample data compared to the fitted one. Sorry. So okay, I'll just go over you. So the question is, does it work in real data? So what we did, we took again Purkinje neurons that I've showed before. In this case, they're labeled with a rainbow approach. So this is the workflow, and we compare the metrics, and then this is the profile, how it looks like. And then the question is, well, if you didn't have it reconstructed, would you have obtained the same data? And the question and the answer is yes. They actually are statistically indistinguishable, the two approaches, and we were able to perform these at a much faster pace. So the other question is, okay, how sensitive it is? Can it be used to resolve topologies that are very similar to each other? And so in this case, I'm showing you now a different data set. So these are probably interneurons. So these cells are very similar, but they are functionally distinct. And so in this case, they're intercellular field. This is the recording pipetting that is injecting the dye in the cell body. and you cannot distinguish the cells by naked eye. And so in this case, we decided to take an unbiased approach. So we, again, this is the workflow. We batch process these images, and then we perform the principal component analysis on all the metrics that we obtained. And we used it. In this case, we used the first component that pretty much had roughly 72% of all the variation and cluster the cells. And what we found is that we were actually able, to correctly identify between 80 to 90% of the times the identity of the cells with just one misassignment in each group. And the reason for that, if you are interested, is actually when we convert the data back, we unbias it back again and compare it to the regional identities. Is that the type 1 cells, predominantly interneurons, have an ascending axon that innervates upper layers in the cortex, and these are these coaxial transaminar axons that you can see here from the reconstruction, although you wouldn't see it. It wouldn't be obvious from the regional images, and we think the plugin is picking up on that shoulder there, which is sufficient to pick the analysis. And so, as a summary, what we think that the plugin accomplishes, that it's a time-saving alternative that can bypass manual tracing. It's accurate when benchmarked against such reconstructions. It standardized the technique, and I think this is the most important. It makes it accessible to anyone. Curve fitting is effective at mitigating segmentation artifacts. Metrics can be used to classify highly related subtypes, as in the case of probable mini-neurons I show you. And I also wanted to point out that it's actually not only applied to neurons, it can actually be applied to several structures, and there's good examples on the literature already that use the plugin to do so, such as memory ducts, retinal vasculature, endogenic sprouting, root morphology, etc. So I just wanted to really finish now just with this idea that what else can we do? We should be thinking about other tools for ImageJ that would be able to allow us to extract morphology direct from tree-like structures without having to do reconstruction. I'm just going to show you some examples. So another approach that we can use to classify the hierarchical branching of orbits called Strauer analysis. You can read that about on the website. And the idea here is to use basically iterative pruning from skeletonized structures to obtain their hierarchical relationships. And there is already a prototype online that you can test that is distributed for the bar update site. And then the other example, okay, I'll finish then. This will be another example. Just want to acknowledge everybody. So everybody that participated in the project and everybody in the ImageJ community that made this possible. Sorry for the. Yeah. Okay.